lapel.
Polymerase chain reaction. Welcome to our YouTube video on the fascinating world of polymerase chain reaction, or PCR. In this video, we will explore the development, methodology, and applications of PCR, all while providing clear and concise information. So, let's dive in. PCR, developed by Kary Mullis in 1983, has revolutionized the field of molecular biology. It all began as a way to remove and use dNTPs from a sequencing reaction mix, but it quickly grew into a powerful technique for amplifying DNA. The methodology of PCR involves the amplification of DNA sequences using a DNA polymerase. By repeating a series of heating, cooling, and extension steps, the number of DNA copies exponentially increases. To perform PCR, you'll need a DNA sequence for amplification, a pair of suitable primers that flank the target sequence, a suitable DNA polymerase, and a supply of the 4 dNTPs. Initially, PCR was done using water baths at different temperatures. However, now we have programmable thermocyclers that automate the process, making it more efficient and precise. Slide 5. The design of primers for PCR. When designing primers for PCR, it's important to consider factors such as primer length, base composition, and the 3N stability. These factors ensure accurate and specific amplification of the target sequence. The most commonly used DNA polymerase in PCR is Taq polymerase, which is derived from a thermophilic bacterium. Other DNA polymerases, such as TFL, TTH, and PFU, also have their own unique features and uses. Reverse transcriptase PCR, or RT-PCR, allows us to amplify mRNA sequences. This is useful for studying gene expression and determining the amount of mRNA in a sample. Competitor RT-PCR is a technique used to determine the amount of mRNA in a sample. By adding a known amount of competitor RNA, we can estimate the target mRNA levels by comparing the amplification products. Nested PCR is a method used to reduce contamination and amplify specific target sequences. It involves using 2 sets of primers, with the second set lying inside the first set, resulting in a shorter product. There are several other PCR techniques, such as inverse PCR, IPCR, and random amplified polymorphic DNA PCR, wrapped PCR. These techniques have their own unique applications and advantages. After PCR, gel electrophoresis is often used to verify the fragment size and assess product purity. Additional techniques like blotting and hybridization can also be used to identify specific sequence regions. PCR has a wide range of applications, including cloning specific sequences, forensic DNA fingerprinting, and diagnostic procedures for genetic disorders. Its versatility and accuracy make it an invaluable tool in the field of molecular biology. The methodology of PCR is quite simple. Denatured single-stranded DNA sequences are copied by a DNA polymerase, creating duplicate double-stranded DNA. By repeating this process multiple times, we achieve an exponential increase in the number of copies of the starting sequence. To perform PCR, we need a few essential components. Firstly, a pair of suitable primers, one for each strand of the DNA duplex. These primers, usually around 20 bases long, flank the target sequence and require prior knowledge of the DNA sequence. The operational sequence of PCR involves 3 main steps. First, the DNA is denatured by heating it to around 95 degrees Celsius. Then, the primers anneal to the DNA by cooling it to about 50 to 60 degrees Celsius. Finally, the DNA polymerase extends the primers by adding complementary dNTPs at approximately 72 degrees Celsius. Initially, PCR was done using water baths at different temperatures. However, now we have programmable thermocyclers that automate the process, making it much more efficient. It's important to note that PCR can be performed using a very small amount of DNA, but precautions must be taken to avoid contamination. The design of primers is crucial for successful PCR. Primers can be obtained from commercial sources or synthesized to order. Factors to consider include primer length, which should be long enough for stable hybridization and uniqueness in the genome. The base composition should avoid repetitive and single-base sequences, as well as internal complementary sequences or overlaps with other primers. The DNA polymerase used in PCR is a key component. The most commonly used is Taq polymerase, isolated from the bacterium Thermus aquaticus. Taq polymerase is thermostable and can add around 1,000 bases every 10 seconds. Other DNA polymerases, such as TFL, TTH, and Fu, also have their own unique features and applications. PCR can also be used with mRNA templates in a technique known as reverse transcriptase PCR, or RT-PCR. This involves first synthesizing cDNA using an oligo, DT, primer, followed by PCR using primers for the target gene. Nested PCR is a technique used to reduce contamination in PCR products. It involves using two sets of primers. The first external primer set generates a normal PCR product, while internal or nested primers, lying inside the first set, are used for a second PCR reaction, generating a shorter product. The applications of PCR are vast and diverse. It can be used to clone specific sequences, clone genes from one organism using priming sequences from another, and even in forensic DNA fingerprinting. PCR is also widely used in diagnostic procedures, such as examining body fluid stains or screening for genetic disorders. And there you have it. PCR is a powerful tool in molecular biology that has revolutionized the way we study DNA. We hope you enjoyed this overview of PCR. If you have any questions or suggestions for future videos, please leave them in the comments below. Don't forget to like and subscribe to our channel for more exciting content. Thanks for watching.
Pharma